Hello everyone! Welcome to the Gen Bio Sisters.
Our group consists of me, Natasha,
Carlene, Cherry,
Anya,
and Gaby.
Today we will be learning about the sexual life cycle and meiosis. Are you ready? Let's get right into it.
As we all know, organisms need to reproduce for their population to survive, and a large proportion of multicellular eukaryotes will achieve this by sexual reproduction.
Unlike asexual reproduction in bacteria, which produce genetically identical offspring, every single offspring from sexual reproduction are unique both genetically and phenotypically.
This is because sexual reproduction involves two parents who will each contribute to half of the offspring's chromosomes.
Sexual reproduction begins with the egg cell and sperm cell, also known as the gametes. Gametes are haploid, meaning they only contain 23 chromosomes out of the 46 in a diploid cell.
And when these two gametes fuse together, they form a diploid zygote which will develop into new organism.
You might ask, how are haploid gamete cells made? Well, this is achieved through a process called meiosis.
Meiosis is the process of converting a diploid cell into haploid daughter cells. They are genetically different from each other. This process will be divided into two parts, meiosis 1 and meiosis 2.
Let's start from meiosis 1. The main purpose of meiosis 1 is to separate homologous chromosomes, and it is basically the first round of division.
Before the actual dividing process of meiosis 1, cells will undergo interphase first. This process contains three phases: G1, where the cells grow in size and make necessary proteins, S, where DNA replications take place, and G2, where cells check for complete components for meiosis.
After interphase, cells will go through prophase 1, metaphase 1, anaphase 1, and telophase 1. In prophase 1, chromatin fibers condense into homologous chromosomes which will form pairs and line up next to each other.
However, during prophase 1, something else occurs. Remember the pairing of chromosomes? Well, the same section of the non-sister chromatids of the pair will be broken off by specific proteins. Afterwards, these parts are exchanged. This process is called crossing over. The point where the chromosomes cross over is called the chiasmata. So, as you can see, crossing over is one of the reasons for genetic variation during sexual reproduction.
Next is metaphase 1. The homologous chromosome will then line up at the metaphase plate with each chromosome molecules of the bivalent located at either side of the equator. Here, centrosomes will have migrated to the opposite poles and spindle fiber are formed already. So after that, the spindle fiber will also attach to the kinetochores of the chromosomes.
Metaphase 1 is followed by anaphase 1. This is where the bivalent chromosomes are separated. This happens because the microtubules attached to the kinetochore shorten, and this causes the bivalent to be separated into two individual chromosomes. The individual chromosomes are pulled towards the opposite poles of the cell, which is also elongated by non-binding microtubules.
Finally, the last step of meiosis 1 is telophase 1. In this process, the nuclear membrane starts to form around the daughter chromosomes at each pole of the cell. At this point, the daughter cells contain half the amount of original chromosomes, from 46 to 23 chromosomes, and the resulting daughter cells are haploid. After meiosis, cytokinesis follows where the cell is completely divided into the two daughter cells.
Now that we have two haploid daughter cells, meiosis 2 can take place. Note that there is no interface between meiosis 1 and meiosis 2. This is to avoid further DNA replication.
The steps in meiosis 2 are very similar to the ones in meiosis 1, which are prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis.
In prophase 2, a similar process occurs. The nuclear envelope breaks down, spindle fibers form, and chromatin fibers in the cell condense into chromosomes. But, since they are haploid cells, only 23 chromosomes will be present.
This is followed by metaphase 2, where the chromosomes will all line up in a single file along the equator of the cell.
After metaphase 2 comes anaphase 2. Here, the centromeres of the chromosomes will divide and individual sister chromatids are pulled towards the opposite poles of the cell.
After that telophase 2 occurs, Here, the nuclear membrane reforms around each group of sister chromatids. Then cytokinesis occurs, which will divide the cell into two completely separate cells.
And so the meiosis is complete. We went from a diploid cell into 4 haploid cells that are all genetically unique.
You might wonder how this is significantly to the sexual life cycle. Well, in human somatic cell, there are 46 chromosomes, which is actually the set of 23. This is the diploid number.
However, for reproduction to occur, the egg cell needs to be fertilized by the sperm cell. And if meiosis did not occur and make the diploid somatic cells haploid, the resulting daughter cell will have 92 chromosomes, which is way too much.
This is why haploid gametes are very important. Gametes are the only cell that is not produced by mitosis in the human body. Gametes are developed from germ cells in the gonad. Egg cells develop in the ovaries for females, and sperm cells develop in the testes for males.
After the fertilization of gametes into zygote, mitosis will occur, developing the zygote into an actual organism by multiplying and specifying the daughter cell's roles.
What makes sexual reproduction so unique is the variation that occurs in every generation. The genetic variation is caused by the independent assortment of chromosomes, crossing over, and random fertilization.
Independent assortment occurs when the homologous chromosomes align randomly during metaphase 1. So when the chromosomes are separated during anaphase 1, the combination of chromosomes will be random.
Crossing over occurs during prophase 1. The contents of the chromosomes are switched with another, and this would also cause the random combinations of traits or genes. Did you know that humans undergo 1 until 3 crossover episodes per chromosomes on average? Imagine how different each gamete would be.
Fertilization will also occur at random. For example, in humans, any sperm can fuse with any unfertilized ovum.
So to put it in perspective, a zygote with about 70 trillion diploid combination is produced from the fusion of two gametes, each with 8.4 million chromosomes combination possible from the independent assortment.
That's all from us for today.
Hope you all learned something new about meiosis!
See you next time!